Hi, this is Miss Litton, and I'm doing an APBio review for chapter 23 and 24 that you did this as a hyperdoc, yes?
Yes.
Okay, so I want to hit, did I start it? I want to hit the parts that maybe you didn't understand, so we'll go over that part. Green algae, common ancestor to land plants. You don't have to know every step of evolution anymore for the exam, but you have to understand maybe some big ticket items like how did we get, what are some things that plants had to have in order to survive on land? And those are kind of like a stair-step process of understanding like each trait that they acquired. Also, it could be useful in an essay if you wanted to show evidence of evolution over time or the development of a species to move on to land. So chara is what's considered to be the common, the ancestor to all land plants. And like the importance of plants, hopefully you know all of that, right? That's pretty easy, right? Why plants are important for us, because we use it for food. Generates oxygen for us, helps keep us cool. They provide shade. Shade, what does it mean, renew the air? They release oxygen, right? Through, hi AP, did you have a long day today? Yeah. Through what process do they release that oxygen? Photosynthesis. I love you so much. Okay. So to move on to land, here's some of the benefits and some of the challenges for moving on to land. Unfiltered sun. Who did the filtering before? Why did they say unfiltered sun? Where were the plants before they were on land? Underwater. So who was filtering the sunlight? The water. Yeah. Okay. More carbon dioxide because you have O2 and CO2 is dissolved into water, but you don't have it at the strength that you would have on land. Nutrient-rich soil, so you have a source of things like nitrogen, which might be deficient in the water that you have. Originally on land, there were a few herbivores or pathogens that you had to worry about. In your hyperdoc tomorrow, you're going to be looking at some of the really kind of cool, I think, plant adaptations for resisting herbivores and pathogens in their immune system. But initially, very few herbivores or pathogens. Challenge of being on land is no water. And water did something besides having water that you need for photosynthesis. Think about when you go to the beach and you see seaweed. Is the seaweed standing up tall? No, it has no secondarily thickened walls. It's just laying there on the beach. And if you lift it up, it'll go back to laying there on the beach. It doesn't have a way to hold itself upright. What is the benefit of holding yourself upright? You can have your leaves up to the sun. Yeah. So scarcity of water and lack of structural support. Those types of things had to evolve for a plant to move on to land. And if we look at this, oh, I know what this is. What is this a picture of? It's a cladogram. Okay, so we're assuming that from here up, they all have what? Roots. Good. Okay. So remember nodes. This is some ancestral species. Do we still have moss today? Yes. Okay. So from here up, moss didn't have this, but from here on up, what do they have? Vascular tissue. What is the vascular tissue that we know and love? Xylem and phloem. How does xylem work? What's the process called? TATC theory, perhaps? Yeah. Adhesion, tension, cohesion. Good. How does phloem work? Pressure flow hypothesis? Exactly. Okay. So the purpose of xylem and phloem is that you could have a source of water near one part of the plant. That source is able to provide water for the whole rest of the plant. Think about plants when they were in water. Every single surface of them could be exposed to water, right? Now they have to have a way to get it. If you're upright, there's no water unless you're standing in a waterfall, okay? They need to have a source of water. So that was a huge hurdle to get over. Okay, next place up is seeds. So having seeds, what did seeds do? They protected who? developing embryo. Good. Okay, and then and your example here is like conifers, gymnosperms, Christmas trees, and Hanukkah bushes, right? Okay, then the next step up is what? Flowers. Okay, and flowers could be used as a what? Attract what? Yeah, pollinators. Now, are all plants pollinated by animals, insects, moths, butterflies, bees? No, some are pollinated by the what? Wind, the air. Okay, so those are not really pretty flowers, but they don't need to be. They don't need to smell good, right? A flower serves its purpose. Remember when we talked about those parasitic flowers that smell like dead, rotting flesh? And so they're pollinated by those insects who tend to like dead, rotting flesh, okay? So it's going to serve its purpose. So these are all anchors in development. Here's another cladogram right here that kind of shows you the different ones. Here's your green algae, okay? So here's the origin of land plants. Bryophytes are those mosses. They're very low to the ground. And then here we have early vascular plants. These would be like ferns right here, and then gymnosperms, and then angiosperms. Now we know it doesn't show you right here in this picture, but the angiosperms are broken into two groups, which we have studied. What are the two groups? Monocots and eudicots. Another name for eudicot is dicot, and that's the old school name, which I tend to go to, but the newest version of it is you're going to call it a eudicot, okay? So, evolution of land plants. Look right here before vascular tissues. What's right here? Protecting the embryo, having some sort of cells around it, okay? So protecting the embryo, then vascular tissue, then seeds, then flowers. That's a really good picture I made. Yes?
Could you just go over the difference of monocots and dicots?
I am. It's coming up. Yeah. Absolutely. Okay. So if you go through this presentation, because it's longish, you can go through each of those steps that I just went over. All right. Next key thing is alternation of generations. Do you and I do alternation of generations? We do not, okay? Our whole body is diploid. Our ovaries or testes are still diploid. But within our ovaries and testes, we have an area where we do meiosis to make our gametes. Plants could have a whole variation. They could have a whole life cycle. There could be one whole plant. You could be swimming next to some seaweed in the ocean, and it could be a diploid seaweed, and you could be swimming, and there's a haploid seaweed, and they look exactly the same to your eyes. You're like, I can't tell the difference. Are you diploid or you're haploid? As plants moved onto land, the trend that you would see, the pattern that you would see, is that the gametophyte is dependent upon the sporophyte, and it becomes smaller and smaller and smaller, to where it's just a few cells, 7 cells to 3 cells big. And it lives and is dependent within the sporophyte structure, okay? That would, yeah. Well, there's so many ways I could go there. An exception to the rule, okay? The exception to the rule, let's go back to these ferns, okay? The unusual thing about the first plants on land, okay, like mosses, let me go back to mosses, their green leafy structure is actually the haploid tissue. And the sporophyte grows out of it like a stalk and goes and releases spores. So that doesn't fit into what we've said so far. Okay, that's the only land plant where the green leafy part is not sporophyte tissue. In ferns, their gametophyte is so small, it's like the shape of a heart, and it's small. You could visibly see it with your eyes, their gametophyte. And the reason why ferns have to reproduce in moist areas is because their gametophyte is like this little kind of unprotected little, very small heart-shaped structure. And then as we move on, when you look at flowers, and if you dissect it into, there's the stigma, the style, the ovary, and as you terrible picture, really terrible picture. Okay, so this is the pistil, the female part of it. Within this pistil, there can be several of them. I'm drawing it obnoxiously big, but it's a 7-cell structure, which is the female gametophyte, and in the pollen on the stamen that contains the pollen, then the pollen itself is going to be part of the male gametophyte. So it's very small and housed within the flower. So that is the trend. Let's talk about what this means. You know what diploid means. You have your homologous pairs, right? And so the structure that on land tends to be the largest structure, the sporophyte structure, within a certain region of the sporophyte, they will do meiosis and make haploid spores. And you just remember, sporophytes always make spores. Okay, sporophytes always make spores. Sporophytes always make spores. This spore is haploid. It will do mitosis. Remember mitosis, you keep the chromosome count the same. You're making a clone. And the cells that it makes is it makes the gametophyte structure. This gametophyte, like I said, might be 7 cells big. It might be as large as a whole seaweed. It might be the leafy portion of a moss. And then that gametophyte does mitosis to make the actual gametes. Then those gametes are going to get together through fertilization and form a diploid zygote. That zygote will do mitosis to grow up into the larger, usually on land, larger, more obvious structure, which is the sporophyte. And I think we've talked about this one other time besides this chapter. Is it somewhat familiar to you? I want you to be very familiar with it. Okay? Sporophyte always makes spores. Gametophytes always make gametes. There are only 2 diploid structures on this circle. What are the 2 diploid structures? Sporophyte and the zygote. Yeah, the sporophyte and the zygote are the only diploid structures on here. Yes?
What exactly is the difference between the sporophyte and sporophyte?
Oh, okay. So you could have like a plant, right? You could have some plant, and it is referred to as the sporophyte, but on the underside of this leaf is the sporangium that actually does the meiosis to make the spores. And spores can be homosporous, like this one is, they all look the same, or they could be heterosporous, where the male and female spores look different. Yes?
So spores are like tiny particles that they're on the plant and they get released?
Yes.
And then there's, how do they get fertilized? Is there like
Okay,
seeds
so
all around?
when we, in the most, like when we talk about flowers, remember when I drew that really like bad picture wherever it is here? Okay, so this whole thing is the sporophyte. It's making a spore, actually makes megaspores and microspores. Microspores here in the male part, megaspores here for the, they do all of that business in here and don't release their spore out anywhere. They keep it housed all the way up into that point. They house it and protect it. You know, it's more protected, right? Just like it keeps it contained until the pollen has to grow a pollen tube, which we learn about not in our next chapter, but the chapter after it. The pollen tube will grow down and fertilize the female. So that way, think of it like internal fertilization. How much better that is than external fertilization, so you're not like, and hope you get together, right? Here, it's maintained inside, and you got to work your way, you got to want it, and get here and fertilize this egg.
So the whole, everything.
Sporang, yeah, all of that contained within it.
And then the pollen gets carried by either the wind or
Or an insect, to deliver it.
And then it kind of like lands on the spore, like near the
It lands on the sticky stigma of the pistil. Here, repeat after me. The female picks up the pistil and says, "Stay, man." Okay, do that whole thing. The female picks up the pistil and says, "Stay, man." The female part is the pistil. The male part is the stamen with all the pollen on it. And so the pollen will either be wind pollinating or it'll be pollinated with a bug who will bring the pollen, the male gametophyte, okay, or gamete, to the sticky stigma, and then they start growing a pollen tube down to get to the, just like they would have to swim through the vagina and the cervix and all the way up, and to get here, they have to grow down and build it and get around there and be the first one in. Very similar patterns. Life. Okay, but we'll learn that stuff where we apply it to a flower. We'll learn that next. Not the one you do without me tomorrow, but the chapter after that. Okay. So this is a simplified picture right here, just like the overall, so you learn the different parts. Then this is showing you an example using fern. So depending on where you are developmentally is how you're going to go about doing this cycle. So for a fern, they're going to have on the underside of their leaves, they're going to have their sporangium, which makes their spores that they release, and they are homospores, and they make this heart-shaped structure. The male parts are right here in the antheridium. This part right here is called an archegonium, and they have to have enough water that they can swim from here to here to fertilize it, and then the new fern will develop up out. When they show these gametes, it's gamete. This guy right here, the zygote, they are developing right out of this part of that heart-shaped structure to make another fern. So, bye, thank you. This will vary. It's done a little bit differently in a moss. It's done a little bit differently in a flower, but you have the same idea as that you have a gametophyte structure and you have a sporophyte structure. But the trend is that as you move, here's my trend one. As you move on to land, the sporophyte gains ascendancy and the gametophyte becomes less and less and less. So that's a good picture to show that. Okay, and we can do these practice problems in a little bit. I just want to make sure I go through all of this. What?
You said class lab, we were all like,
I'm ready. Is there anything on that you want me to go over? Okay, so now chapter 24, flowering plant structure and organization. The thing I would pay attention to out of here are just different adaptations that plants might have where you look for connections between structure and function, okay? So for instance, this is corn, this is called a radicle, it's growing down, it's going to be the root. These are all tiny little hairs. Why would it be adaptive to have root hairs? More surface area for water, primarily water absorption, okay? So this is like, no. So this is where I think you were getting in, because water is a nutrient, right? Just not a mineral or anything like that. But yes, it would hurt. So this would increase water absorption. Understanding when you look at a tree, the bulk of what you're looking at, this is all xylem. Do I have this be anything? Oh, I do. Woody stem. Okay, this is all xylem right here. Remember how I talked to you how the xylem, it keeps getting made to the inside of the vascular cambium and the phloem to the outside. And so the vascular cambium that can, in dicots, which can have secondary growth, the vascular cambium would be right there, and they keep making more xylem to the inside and more phloem to the outside. You have prop roots to help support it. You know what those are. Those are leaves, and we'll talk about that in just a second. Okay, so when you look at angiosperms, okay, when we look at flowering plants, I want you to kind of look what happened here. So here was green algae. These are outgroup. And from here on up, what do they have? Embryo. So first you protect your embryo, right? Then you have vascular tissue, xylem and phloem going both ways, right? Then you have seeds, and then you have flowers. Yes?
if you go back to the picture with the wood stem, would you say that the middle xylem is the newest one? Like the one closest to
The closest to this is the newest.
Oh, okay.
This would be called like heartwood. It's probably all filled with sap and doesn't work. And this would be called sapwood. Okay, so your green algae. First thing we want to do is what? Protect. Do this. Protect your embryo. What am I protecting? What am I holding here? A baby. Good. Then you have xylem and phloem, right? You have vascular tissue. So you're here, and you just lift it up. Now you have xylem and phloem. Then you have seeds to protect, right? And then you have a flower. Okay, so there you can see the different leveling up. So flowering plants live in a lot of habitats, tiny, small. Ovules always enclosed within diploid tissues. Okay, diploid tissues. So the ovules are what's going to generate the egg, and they're surrounded by diploid tissue. So that means they're surrounded by sporophyte tissue, right? Does that make sense? Okay. And the ovary develops into fruit. When you eat fruit, like when I eat an orange, I think I've already had an orange today. When I eat this orange, not the skin, because ick, but inside, I'm eating the orange tree's ovaries. And you remember when I said the female picks up the pistil and says stamen? Remember that? The pistil at the base of it, she actually has carpels, and that's why there's sections of orange, because she has multiple babies in each section. It's like having uterus, where you go like this all the way around in a circle. Yeah. So next time you have fruit salad, you're going to say, I'm having plant ovary salad. Yeah, I love plant ovaries. It's so good. Okay. So you do not need to know all the plant tissues, though that's super clever that I found Kleenex that had plants on them. You don't need to know all of that part anymore. But when you see something that says meristematic, what do you hear in there that you know about? Stem cells. Okay, so stem cells are embryonic cells that can be used for multiple purposes, like in your bone marrow. Plants, remember we talked about this. Mitosis for us is for growth and repair, right? Okay, and I said I lose the ability. I can't grow. Remember, my growth plates have stopped being growth plates. So plants always have meristematic tissue. Okay, it's just where it is located. And that is one of the big differences between monocots and eudicots, because monocots have their marrow, you can think of like their stem cells are originally laid down and they can't make any more. So all that monocots can do is grow straight up, like a blade of grass, like corn, like a palm tree. Just grows straight up. Whereas eudicots have what's called a vascular cambium, and they can continually put more xylem to the inside and more phloem to the outside, so they can branch and spread out and usually live for multiple years. Whereas most monocots are just annuals. They live 1 year. Exception, palm trees. Okay? All right, so that's meristematic tissue. Don't worry about that. They're never going to ask. Okay, there are meristematic tissues for the epidermis layers, the ground cells, parenchyma, collenchyma, sclerenchyma, and vascular tissue. Vascular tissue, when you think of vascular tissues, what do you think of? Xylem and phloem. Yeah. So procambium makes those. Parenchyma, collenchyma, and sclerenchyma. I'm just going to tell you right off the bat here. Parenchyma, if you were like to take every plant and put it in a blender and reach in once you turn the blender off and pull out a cell, it's parenchyma. It looks like parenchyma, okay? But most cells are made out of parenchyma. Collenchyma are like edges of leaves, help it be solid. When you eat celery, you're eating sclerenchyma. I'm sorry, collenchyma. You're eating celery. You know, it's crunchy collenchyma, okay? You can still call collenchyma because it's alive. I call you collenchyma, okay? It's still alive. Sclerenchyma, I want you to think about walnuts. If you went to go eat a walnut, okay, and you bought the walnuts, could you like take the shell and just bite it open? No, that's dead at maturity. That's sclerenchyma. Like, when Tarzan swings through the jungle on vines, he's got like sclerenchyma in there. Ooh, you know another place you can find collenchyma? Well, it's okay. All right. You're like, why is she going off on that? This is something you're gonna hear about tomorrow, okay? On plants, yes.
You need to know collenchyma.
I'm just introducing it in case like they have some question where they use it as background and they don't expect you to know it. You're gonna be like, collenchyma, I totally know there's a lot of collenchyma. Okay? Yes.
Didn't it say that phloem were made out of parenchyma or whatever? Is it all the same?
I'm going to go over that. Yes. Sclerenchyma, remember we talked about xylem being dead? Sclerenchyma. And I'm going to give you some more examples. What?
We're just trying to find out if you're eating black licorice or like a purple Twizzler.
Oh, it's a purple Twizzler.
Okay.
Purple Twizzler. Okay. Look, you know what that is. What is that? Stomata. Good. These right here are called trichomes. They could have poisons in them. This would be something you would learn about. This is an adaptation. You need to know about that. But this is an epidermal cell, right? And it's secreting. Have you ever like touched stinging nettle or something and it's hurt you? Like, right? Something that has a toxin on it. Those are usually trichomes. And you're going to learn about those. And here's some more trichomes. You're going to learn about those tomorrow. Here are those 3 cells. Here's parenchyma. Here's parenchyma in celery. Here's sclerenchyma. Walnuts, but you know when you get to that tough part in the center of a pear? Huh. Yeah, sclerids. Now here is your xylem and your phloem. You know about that and those vascular tissues. We had to learn about that in the other chapter. Here's your phloem. Oh, sorry. Yes, xylem. Here's your xylem. Here's your phloem. This is a really good chart because it kind of breaks everything down. What would this mean? Chlorenchyma. Chlorenchyma. What do you think that means?
It's a mix between
Chloro. It might be green and do what process? See? You're so smart. Okay. Now, you know roots, stems. When you look at a shoot like this, what do you think that is? A monocot or a eudicot? Monocot. You know why? Comes straight off. It doesn't have any petiole. Remember the petiole that could hold a leaf? Look at this. See the petiole? That's the petiole right there that's holding the leaf. Now, monocots have what's called, if you just think of grass, think about grass outside in your lawn. It always has those parallel veins, so that's typical of a monocot. Whereas here you have, this is called, what shape does it look like it's in? My palm. That's called palmate venation when it spreads out like that. And there's pinnate venation would look like a leaf that looked like this. Would be like that. Whereas a monocot, I would look more at a blade and it has parallel venation. So that's one of the differences when you look at, first of all, I want to see what this says. Yeah, 2 groups. Okay, so on this chart right here, where the name monocot and dicot came from would be the seed leaf. Remember, monocots and eudicots all have seeds. So do gymnosperms, right? But gymnosperms, right, have seeds. Angiosperms still have seeds, but they have also a flower, okay? So if you take all the flowering plants in their seeds, you either have monocots or eudicots. If you think about a sunflower seed, when you eat a sunflower seed, it's just a solid seed inside. But when you eat a walnut, there are 2 halves to it, right? When you eat a bean and you bite down, there are 2 halves to it, okay? Those are eudicots. They have 2 halves, or dicots. Same thing. Monocot is when it's solid in the center. So that's one way to recognize it. Another way to recognize it is if you look at the roots. Look at the roots. Who has the star? Yeah, the eudicots. You know what that's called. What's that called? This is the epidermis. This is the endodermis, right? And it has the Casparian strips around it. And this is the stele in the center. Remember we talked about the apoplast and the symplast? Okay, so when you have a star like that, you know you're talking about a eudicot. When you look here at a monocot, their veins, their vascular bundles are scattered. But when you look at a eudicot, where you see this right here, see the red and then the blue? The red is xylem and the blue is phloem. So it just keeps making more red xylem to the inside and phloem to the outside. Okay, and this right here, this part right here, between the xylem and phloem that I made really big, is called the vascular cambium. Those are stem cells. And that's why eudicots can have secondary growth, because they always have a source of making more parts. Okay? Then you already know this, venation, monocot, right? This would be pinnate venation of a eudicot. Okay, this is parallel. And then what's kind of interesting is monocots have flower parts in threes or multiples of threes, whereas eudicots are fours and fives. So if you're going to play he loves me not with a monocot, you might want to start with he loves me. Okay, because it's always multiples of 3. So where are you going to run into trouble? Yes, 12. Okay, I know that. So when you count and look at them, that would be another way to, and this is a way to see how structurally it's different between monocots and eudicots. Then if you looked at this right here, that would be a monocot stem because it's got scattered vascular bundles. This is a eudicot stem because I have vascular cambium and xylem to the inside and phloem to the outside. This would be another eudicot. Xylem. It looks like little acorns, doesn't it? Like, okay. And then this is scattered. I think these look like monkey skulls. Monkey monocot, just like monkey face, or aliens a little bit. The eyes are xylem. The forehead is phloem. Just telling you, in case you're wondering. Okay. Are these monocots or eudicots? Eudicots. Good. So you could put it in a basket. Would you put this in the Would you put this in the monocot or the eudicot basket? I agree. How about this one? Oh, that's a hard one. Here, what about this one? Good. What about this one? What about this one? What about this one? What about this one? What is that? 1, 2, 3, 4, 5. Sounds like a eudicot. Now one of these is going to go here and one of these is going to go here. Oh.
Yeah.
Okay, what about this one? So stupid. Let me give you a hint. One of these goes in this basket and the other one goes in the other basket. Okay, what's this one? Go in that basket, monocot. Okay. All right. How are you feeling about monocot, eudicot? Better? Okay. All right. And then there's a bunch of questions. What's this? You good about that?
Yeah.
Okay. All right. And then, no. What do I want here? Oh, this is pretty interesting. Probably you just looked at this and just went away. But there's a lot of stuff in here. One, what does TF stand for? Transcription factor, right? Transcription factors help RNA polymerase. Okay. Blue is, I tried to help you by highlighting things, whether it was genes or a transcription factor. Blue is DNA and green is a protein. So this is taking into plants something you know about controls of gene expression and how that would go about. So you're going to be learning about day length, short day plants, and long day plants. You're going to learn about that tomorrow. Can I just give you a little hint on something, which you will see? Short day plants, they learned it doesn't have anything to do with the length of the day. It really has to do with the length of the night. So if I'm a short day plant, which means I flower when there's a short day, I'm really flowering because there's a long night. And if I'm a long day plant, I'm really flowering because there was a short night. Okay? Really has to do with the night. So this is showing you about appropriate day length allows the accumulation of the transcription factor, which I haven't highlighted, CO, that controls expression of the FT in the gene. FT transcript moves through the phloem to the shoot apex where the FT protein, so transcription factor stimulates this gene so you get this protein. Okay, the complex then activates another gene. Okay, so this right here, this FT protein produced and interacts with the transcription factor, protein with the transcription factor. This would be like an inducer, right, on that transcription factor. So it can affect AP1, that's a gene, starts flower development, but it also needs this transcription factor leafy, okay? And then as a result of that, you get your flower. So it's a combination of transcription factors affecting genes, which then affects flowering. So, yeah, that's kind of complicated, but you could get questions like that, okay, where they ask you to put it all together. That would be a good question because you have to understand about DNA, gene expression, and then control of control that you have over that gene expression. Okay, then here it talks about roots and looking at roots, and this is a structure function. Zone of cell division, this is where you would see mitosis taking place. Zone of elongation, any guesses there? The cells get longer. And then zone of maturation, notice what section has root hairs? Zone of maturation. Because those root hairs are very fragile, and if you're still growing down through and elongating, your cells are moving when you're making more cells, or elongating, you're moving, you could rip the root hairs off. So you wait till you're maturing and it's not moving anymore through the soil, then you have your root hairs. Does that make sense? Okay. Look this right here. There's a star in the middle. So is this a monocot or a eudicot root? Eudicot. Yeah. Oh, plus I labeled it eudicot root. Here would be the difference between a dicot or eudicot root and a monocot root. You always have the star in the middle of the other one. Notice you still have endodermis, you still have pericycle, it's just what's in the center. You have the xylem in the center of the dicot root. And then these are just some pictures of that. The vascular cylinders. This is showing you root hairs. This is a review of the apoplast and the symplast. Okay, the Casparian strips around the endodermal walls prevents water from moving any more between the cells, which is called the apoplast, and it forces it to be regulated through the symplast. Good, good. That just labels those things. Root adaptations. So roots anchor the plant, absorb nutrients, and they can store nutrients as well. And then these are just some different kinds of roots for storage. Pneumatophores, these plants, the roots grow down and then back up because there's no water in in the swamps, and so they have to get their oxygen. They have to extend parts of themselves up there. And then those are just other ones. Those are stems. Stems have to do what that are brown? Cellular respiration or photosynthesis? So what gas are they going to need? Oxygen. How are they going to get it? Just like leaves have stomata, stems have what's called lenticels to allow oxygen to get in. And then this is it, just showing you different kinds of leaves and stems, stem modification. You did a hyperdoc last time on those curling tendrils. There are leaves, leaf venation, photosynthesis. You don't need to know that. Leaf modification, and you're done. Good job, you guys. 2 chapters. Boom. Proud of you. Yes.
Do you eat kiwis?
What?
Do you eat kiwis? Like the fruit?
Yes, kiwis.
Do you eat them with the skin or without the skin?
Without the skin.
What?
Of course without the skin.
Yeah, the skin is like
You're welcome.
furry.
Study hard. Yeah, I wouldn't eat that. Have good evenings.